We are pleased to have Dr. Deanna Kroetz give today's lecture. Deanna is a professor in the Department of Bioengineering and Therapeutic Science in the School of Pharmacy at the University of California, San Francisco. She received her BS in Pharmacy from Ohio State University and her PhD in Pharmaceutics from the University of Washington. Deanna was a Pratt's Fellow at the NIH in the Laboratory of Molecular Carcinogenesis in the National Cancer Institute before joining the faculty at UCSF. She has received numerous awards over the years, including AAPS New Investigator Award in Pharmacokinetics, Pharmacodynamics, and Drug Metabolism, the Leon Goldberg Young Investigator Award from ASCPT, a Fellow of AAPS and AAAS, and a Distinguished Alumni Award in Excellence in Pharmaceutical Science and Research from the University of Washington School of Pharmacy. Her research interests are focused on understanding the mechanisms underlying inter-individual variations in drug response and toxicity. Deanna served as the Director of Pharmaceutical Science and Pharmacogenetics of their graduate program at UCSF from 2010 to 2019. She is currently the Deputy Editor-in-Chief of Clinical and Translational Science. Please enjoy today's lecture.
I'm Deanna Kroetz from the Department of Bioengineering and Therapeutic Sciences in the Schools of Pharmacy and Medicine at the University of California, San Francisco. And I'm going to be talking to you today about phase 2 metabolism. So I'll start with just an overview of the characteristics of phase 2 metabolism, followed by more details about 3 specific conjugation reactions: glucuronidation, sulfation, and glutathione conjugation. And then I'll end with a summary of what I've covered. So just to orient everyone to what we're going to be talking about today, so drugs and other xenobiotics can be metabolized to phase 1 metabolites, largely by the cytochrome P450 enzymes, and this will be covered in a separate lecture. But these xenobiotics and drugs can also be conjugated to what is referred to as a phase 2 metabolite. This can be either a direct reaction with the parent drug, or it can be conjugation of a previously formed phase 1 metabolite. And these phase 2 metabolites then are eliminated from the body, either in the urine or in the bile. So this is an interesting analysis of about 1,200 substrates where they looked at the sequential metabolism of these substrates and captured all their metabolites, looking at about 6,800 distinct metabolites. And on average, this was about 6 metabolites per substrate. And what they found is that the initial metabolites that were formed were largely phase 1 metabolites. About 70% of these initial reactions were catalyzed by phase 1 enzymes, mostly P450s. About 10% were hydrolytic reactions, and a little over 20% were direct phase 2 conjugation reactions. But as these initial metabolites became further metabolized, conjugation reactions or phase 2 reactions become quantitatively more important. As second generation metabolites, they represent about 40% of the metabolites that were formed. And as these metabolites are metabolized even further to third and further generation metabolites, they approach about 50% of the metabolites that are formed. So for a phase 2 reaction, functional groups on the drug or xenobiotic interact with endogenous substrates. And they do this through the use of activated cofactors. The conjugation reactions that comprise phase 2 reactions are glucuronidation, sulfation, acetylation, methylation, amino acid conjugation, and glutathione conjugation. And we'll talk about a couple of these in more detail. The main purpose of these phase 2 reactions is to increase significantly the hydrophilicity of these metabolites, which will then facilitate their elimination. And typically these conjugation reactions lead to elimination of the conjugates into both the urine and the bile. Quantitatively, glucuronidation and sulfation are the most important conjugation reactions for drugs, and glutathione conjugation is very important for eliminating highly reactive electrophilic metabolites. So these phase 2 reactions occur at nucleophilic sites in drugs and xenobiotics. And conjugation at these nucleophilic sites by glucuronidases, sulfatases, and amino acid conjugating enzymes leads to an increase in water solubility that will facilitate their elimination from the body. And conjugation at these nucleophilic sites leading to N-acetylation or methylation increases the nucleophilic reactivity of these conjugates. In addition, conjugation can occur at electrophilic sites on drugs and xenobiotics, and these conjugates that are formed eliminate the reactivity of these electrophiles and they increase water solubility. And this is glutathione conjugation. This is just a summary of the conjugation reactions that occur. What's interesting here is, unlike the phase 1 reactions, which largely occur in the endoplasmic reticulum in the microsomal fraction of the cell, most of the conjugation reactions occur in the cytosol, with the exception of glucuronidation, which also occurs in the endoplasmic reticulum. There are specific enzymes that are needed to catalyze each of these reactions, as well as specific cofactors. And we'll talk in more detail about a few of these reactions. Quantitatively, the most important conjugation reaction is glucuronidation, followed by sulfation, and then glutathione conjugation. And so those are the three that we're going to talk about in more detail. N-acetylation and methylation, amino acid conjugation, they can occur on drugs, but quantitatively, they're much less important. So we will start with glucuronidation. And glucuronidation can occur on a nucleophilic oxygen, which is represented here, but it can also occur on a nucleophilic nitrogen, sulfur, or carbon group. The cofactor for this reaction is uridine 5'-diphospho alpha-D glucuronic acid, or UDPGA. The structure is shown here. And this glucuronic acid moiety at this end of the molecule will react with the drug to form this glucuronic acid conjugate. This alpha-D glucuronic acid forms a beta glucuronide through a backside attack. And these glucuronide conjugates are mostly charged at physiological pH with a pKa of about 3 to 4. This is catalyzed by a glucuronosyl transferase enzyme. These enzymes, the UDP-Glucuronosyl transferases, are referred to as UDPGTS or UGTs. These are microsomal proteins, the only conjugation reaction that occurs in the endoplasmic reticulum. As I just mentioned, the cofactor is UDPGA. And importantly, these are low affinity enzymes, but they are high capacity. And that is why they are quantitatively the most important for phase 2 metabolism. And glucuronidation is typically the most common phase 2 reaction that you'll find. There are 17 human isoforms of the UGTs, And the most important for human drug metabolism are UGT1A1, 1A3, 1A4, 1A6, 1A9, and UGT2B7. This is some data from Bhagwat Prasad's laboratory at Washington State University, where they have used proteomics to quantify the protein level of the UGTs, both in the liver as well as in the intestine. And you can see a different distribution of these UGTs in these two tissues. So for example, in the liver, UGT2B7 is the most abundant isoform, followed by UGT2B15, where in the intestine, UGT2B17 is the most abundant isoform. It represented a very small fraction of what was present in the liver. And this is important for understanding which of these isoforms are going to be more important for glucuronidation in the liver and intestine. Here's an example of the most common glucuronidation, which is glucuronidation of oxygen groups. And this is an example with codeine and morphine. So codeine, shown down here, can actually be directly glucuronidated by UGT2B7 to the codeine 6 glucuronide metabolite. It is also in a minor pathway, phase 1 reaction catalyzed by CYP2D6 converted to morphine, which is highly responsible for the analgesic effect of codeine. And morphine, in turn, can be glucuronidated at both the 3 position as well as the 6 position to form morphine 3 glucuronide and morphine 6 glucuronide. And you can see these are both catalyzed by the same UGT enzyme, UGT2B7. And the major pathway is the formation of morphine 3 glucuronide, with only a minor amount going to the 6 glucuronide. Another example of O-Glucuronidation is with bilirubin. Both of the carboxylic acid groups on bilirubin can be glucuronidated, in this case by UGT1A1. And we find both the bilirubin monoglucuronides as well as the bilirubin diglucuronides being formed. This is an interesting pathway. It's a mechanism for elimination of bilirubin. And this enzyme, UGT1A1, is actually not active at birth. And so in neonates, it takes several days for UGT1A1 activity to be turned on. And that is why in neonates, there can be accumulation of bilirubin that's responsible for jaundice. After a couple of days, UGT1A1 activity is usually sufficient to eliminate bilirubin from these neonates. I mentioned that the glucuronide conjugates that are formed are going to be highly charged at physiological pH. And because of this negative charge, they require transporters to cross cell membranes. This is important for secretion of glucuronides into the bile, especially for larger molecular weight glucuronide conjugates. They can be highly secreted into the bile after they're formed in the hepatocyte. And the 2 transporters that are most important for glucuronide secretion in the bile are the breast cancer resistance protein, or BCRP, and the multi-drug resistance associated protein 2, or MRP2. For glucuronides that are formed in the hepatocyte to circulate in the plasma and be able to, for example, get to the kidney for renal elimination, they also require a transporter on the basolateral membrane. of the hepatocyte, and MRP3 has been shown to be critical for the elimination of most glucuronides from the hepatocyte into the plasma, with some potential contribution from MRP4 as well. For glucuronides that are secreted into the bile, they can also undergo this process of enterohepatic recirculation. This is illustrated here, where you have a parent drug, such as morphine, being converted to the glucuronide. This glucuronide, if it's efficiently secreted into the bile by mostly BCRP or MRP2, these glucuronides then will be dumped back into the intestine, where they can be converted by bacterial beta-glucuronidases back into the parent drug. This parent drug then can be reabsorbed back into the systemic circulation, enter through the portal vein into the liver, and go back out into the systemic circulation. And so what you see with this enterohepatic recirculation is you see a second peak of suggesting that there's additional absorption of the drug at some later time after the Cmax from the original absorption process. And so any kind of later bump in the concentration time curve is an indication that you might have enterohepatic recirculation going on. Here's an example of this beta-glucuronidase reaction. This is with the morphine 3 beta-glucuronide, which can be converted back to morphine. And in this case, morphine could end up back into the systemic circulation again. Glucuronides can also be eliminated from the body in the urine, again, because they're going to be charged at physiological pH. They will require transporters to facilitate this secretion into the urine. And this is a concerted effort of both uptake transporters on the basolateral membrane and efflux transporters on the brush border membrane of the renal epithelial cells. The major uptake transporter for glucuronide conjugates is the organic anion transporter 3, or OAT3, with some contribution from the OAT1 transporter as well. And on the brush border membrane, the most important efflux transporter is MRP2, multi-drug resistance associated protein 2, with both MRP4 and the mates also transporting some glucuronides. Mass balance studies provide information about all of the metabolism that's occurring within the human body. And these quantify the metabolites in plasma, urine, and feces to be able to account for all of the drug and its elimination. From these types of studies, we have evidence that there is intestinal metabolism of glucuronides. Here's an example of a C14 mass balance study of a TGF-beta R1 kinase L5 inhibitor, this LY compound here. And you can see that there are multiple routes of elimination of this compound. And for each of these metabolites, it's indicated whether it was detected in plasma, urine, and or feces. Here's an example of a direct glucuronide being formed from this M7 metabolite. So you get this M7 metabolite, and then the glucuronide of M7 is the M8 metabolite. The M7 metabolite is found in plasma, urine, and feces, but the M8 metabolite is only found in the plasma and the urine. This suggests that the M7 glucuronide form in the intestine was hydrolyzed back to M7, and what you find coming out in the feces is only the hydrolyzed glucuronide metabolite M7. This is similarly shown here for this M3-M4 combination, with the M3 being found in the feces, but the glucuronide of M3, the M4 metabolite, only being found in the urine. So we know that this intestinal metabolism by bacteria of glucuronides is commonly found for a lot of glucuronide metabolites. There can be quite a bit of variability in glucuronidation, interindividual variability that's regulated particularly by various disease states. In this particular example, these investigators were looking at the pharmacokinetics of morphine, as well as the morphine 6 and 3 glucuronide. And they compared concentration time profiles in healthy volunteers on the left to patients, renal failure patients on the right. And what you can see is that the elimination of morphine, as well as the morphine 3 and 6 glucuronide metabolites, is significantly impaired in the renal failure patients. It's been recognized for a number of years that renal failure actually affects hepatic metabolism, particularly phase 1 metabolism and phase 2 metabolism. And so that's what is probably represented by this delayed elimination here of morphine. And then renal failure is also critical for elimination of metabolites. So this long sustained accumulation of morphine 3 and 6 glucuronide likely reflects the inability of the kidneys to secrete these morphine glucuronides as well as they do in healthy volunteers. So in this case, renal failure causes both a decrease in the parent drug metabolism, as well as a decrease in the elimination rate of the metabolites that are formed. There are also differences in glucuronide levels of morphine that have been demonstrated in patients with non-alcoholic steatohepatitis or NASH. In this case, there's no effect in these patients with liver disease on the elimination of morphine. But what you do see is that both the morphine 3 glucuronide and the morphine 6 glucuronide levels are higher in patients with NASH compared to the healthy controls. And in fact, there's a correlation between the morphine glucuronide, this is a combination of both 3 and 6 glucuronide, in either the Cmax or the AUC. There's a correlation with those levels and the severity of the liver disease, which is this NASH fibrosis score. This is actually related to the fact that in patients with NASH, they overexpress the MRP3 transporter on the basolateral membrane of the hepatocyte, leading to higher levels of the glucuronides in the plasma. The mechanism of this increased regulation of MRP3 in NASH patients is not completely understood, but there's multiple lines of evidence that suggest that this leads to increased circulating levels of glucuronide conjugates. In general, we think of conjugation reactions occurring as a way to eliminate the pharmacological activity of a drug, but there are examples of active glucuronide metabolites that have been reported. And the best characterized of these is this morphine 6-glucuronide metabolite. It's the minor glucuronide metabolite formed from morphine, but it has its own pharmacological activities and is an effective analgesic. In fact, in this randomized controlled trial with about 250 patients in each arm of this study, they directly compared the analgesic activity of morphine 6-glucuronide in these solid circles to the standard of care morphine in these open circles. And they compared the ability of morphine 6-glucuronide to give effective analgesia following a major abdominal surgery. So you can see if you look at this insert up here, at early times, the morphine 6-glucuronide does not give as good of analgesia as morphine itself. So there's some delay in the analgesic effect compared to the parent drug morphine, but after that, there's very little difference. And this clearly demonstrates that morphine 6-glucuronide is an effective analgesic. You can see this as well in data looking at patient-controlled analgesia. How many times do they self-administer morphine or morphine 6-glucuronide per hour is what is plotted here on the y-axis. Again, at early times, it looks like morphine 6-glucuronide might not be quite as effective as morphine, but these differences disappear, or if anything, the morphine 6-glucuronide actually looks even more effective at later times past surgery. So we'll now move on to briefly discuss sulfation, another important conjugation or phase 2 reaction. The substrates for sulfation are very similar and usually overlap with those for glucuronidation. Any drug that has a nucleophilic oxygen or nitrogen can form a sulfate conjugate. And this requires the cofactor 3' phosphoadenosine 5' phosphosulfate, or PAPS, which is shown here. And the sulfate group over here will be transferred to the nucleophilic site on the drug to form this sulfate conjugate. This is catalyzed by sulfotransferases, and similar to glucuronide conjugates, these sulfate conjugates are going to be highly charged at physiological pH. The sulfotransferases that catalyze the sulfation conjugation reaction are called salts. These are cytosolic proteins. As I just mentioned, they require the cofactor PAPS. And in contrast to the UGTs, these are high affinity but low capacity. And so what that means is at lower concentrations, the sulfotransferases will be more important than the glucuronosyl transferases. And you'll see more sulfation at lower concentrations than glucuronidation. But as the capacity of the sulfotransferases is approached, glucuronidation becomes quantitatively more important. The major human salt isoforms are noted here. These are salt 1A1, 1A3, 1B1, 1E1, and salt 2A1. As I mentioned, sulfation and glucuronidation often occur at the same positions. This is illustrated here for this NMDA receptor antagonist. you can see that if you look at metabolite 6 and metabolite 19, these are direct conjugation reactions, direct glucuronidation, and direct sulfation of the parent drug at the same position. And if you look at metabolite 7 and metabolite 14, you can see both glucuronidation and sulfation, again, at the same position, occurring on this phase 1 metabolite that was formed by cytochrome P450 2D6, the M13. And so this is very common. These nucleophilic oxygens and nitrogens, you'll typically see a mix of both a sulfate conjugate and a glucuronide conjugate occurring. The last conjugation reaction that I want to highlight is glutathione conjugation. Here is the structure of glutathione. It's a tripeptide, gamma-glutamyl cystenyl glycine. And glutathione is found at high concentrations in essentially every cell in our body, often at millimolar concentrations. And that reflects its important role in detoxifying any reactive electrophiles that might form in our cells. There's actually 2 pools of glutathione within our cells. There's a smaller mitochondrial pool that has a long half-life, but most of the glutathione is found in the cytosol. This is the glutathione that's important for drug conjugation, and it has a relatively short half-life of 2 hours. Glutathione conjugation is catalyzed by the Glutathione S-Transferases, or GSTs. And these GSTs, they function as either homodimers or heterodimers within their class. The major isoforms are shown here. So for example, in the alpha class, you could get homodimers or heterodimers of any of these, catalyzing these reactions. Kind of the classic example of glutathione conjugation being critical for the detoxification of a drug is with the analgesic acetaminophen. The structure of acetaminophen, or APAP, is shown here. And under normal conditions, at normal doses of acetaminophen, you get both a glucuronide conjugate as well as a sulfate conjugate that are formed. A very minor pathway of acetaminophen metabolism is by cytochrome P450 2E1 with some contribution from CYP3A4 to form this, eventually, this reactive metabolite NAPQI. Because this is a reactive electrophilic metabolite, it will be conjugated by glutathione. And this glutathione conjugation will increase its hydrophilicity, leading to renal excretion, and also eliminate its reactivity. However, if part of this NAPQI escapes this glutathione conjugation, NAPQI can actually react with the hepatocyte itself, leading to hepatotoxicity. So it's understood that there's a lot of interindividual variation in acetaminophen metabolism that can influence the risk of developing hepatotoxicity. At normal doses, the majority of acetaminophen will be eliminated through these glucuronidation and sulfation pathways. However, if this is compromised in any way, for example, from genetic defects and glucuronidation with Gilbert's syndrome, then this pathway could be compromised, causing more drug to go through this potentially toxic pathway. Of course, when you have an overdose situation, you also will begin to saturate this pathway, again, causing more of the drug to go through this reactive pathway. You can also have particular conditions such as prolonged fasting, chronic alcohol ingestion, as well as some drugs that are known to induce cytochrome P452E1 that can lead to a larger percentage of acetaminophen metabolism occurring to this reactive NAPQI pathway. And when you have these situations and you have a large amount of NAPQI being formed, you can also deplete your glutathione stores. Even though we have these millimolar concentrations in the cell, they can be depleted under these overdose conditions. You can also have depletion of GSH due to chronic liver disease, as well as chronic alcohol ingestion and under conditions of malnourishment. And so in all of these cases, that would decrease the ability of our bodies to glutathione conjugate NAPQI as a way to detoxify this compound. And then more NAPQI has the potential to cause hepatotoxicity. So let me summarize the major points that were made in this lecture. Phase 2 metabolism is an important mechanism for increasing water solubility and facilitating elimination of drugs from the body, both in the urine as well as in the bile. Most conjugation reaction occurs on metabolites that have been formed through a phase 1 reaction, typically a P450 mediated phase 1 metabolite, although there are examples of direct conjugation of parent drugs, for example, morphine as well. The most important conjugation reaction quantitatively is glucuronidation. This is a high capacity reaction, and this is followed by sulfation and glutathione conjugation. Because the purpose of these conjugation reactions is to increase hydrophilicity, these conjugates typically are charged at physiological pH and require transporters for urinary and biliary secretion. In general, the conjugates that are formed through these phase 2 reactions are going to be pharmacologically inactive, but there are examples, the one that I illustrated with morphine 6 glucuronide that shows pharmacological activity. Glutathione conjugation is our most important mechanism for detoxifying electrophilic metabolites and preventing toxicity of drugs and other xenobiotics. And we are beginning to recognize that there's quite a bit of inter-individual variability in the glucuronidation and sulfation reactions, both in their formation as well as their elimination. And this can be influenced by disease state. It can be influenced by genetics of the enzymes involved, as well as drug-drug interactions.